To Gen Z Speaks. We have a very interesting guest today, Josh McGinnis. I hope I got your last name right, man. Josh is a biotech startup founder and software engineer. He previously worked at Intuit, and now he's starting his company, Every Man Bio, and also has a podcast, which has been really fascinating to listen to. Josh, welcome to the show, man. Appreciate you.
Hey, thanks for having me, guys.
Let's dive right in. Let's dive right in. I want to start with your personal background, since biotech is such a wide field, right? And I feel like there, I don't know a lot of people in this specific field, and because it's so detailed and nuanced, what motivated you or inspired you to be involved in that space?
Well, ironically, I don't have a background at all in biology. Although I'm engineering fungus here in my home lab, I don't have any background in biology. In fact, my background's in computer science. And I spent the last 15 years as a software engineer in a variety of different positions, mostly working in what's called fintech, the financial world, dealing with big data and things like that. But as most people go through some different phases in life, you guys are a little bit younger than I am, but you will go through phases in life where you do something for a while and you realize, you know what, there's so much more that I have to offer to the world. And so I was looking to do something new and different. And I also should mention that I had a very positive and life-changing psychedelic experience, which sort of prompted me to explore the world of biology and fungi in particular. And so about a year and a half ago, I decided to go all in teaching myself genetic engineering, synthetic biology, and with a specialization on fungal genetics. And so I've been on this journey with hopefully I'm going to be the first person in the world to genetically engineer fungus in a DIY home lab, which I'm happy to talk more about. So that's the journey that I've been on, and that's what I'm working on today.
That's fascinating. Hey, Josh, Jenish is going to be asking you a lot of questions about your psychedelic experience for sure. But before that, I want
Yeah.
you to help us understand. And obviously, we have a basic understanding of biotechnology, but for the sake of the audience, biotech has different types of subfields. Like there's agricultural biotech. There's biotech is being used in all sorts of different arenas, basically to help improve life. But for you, in your opinion, how do you define biotech and the space that you're working on? How would you, if you can just help us understand the areas of research that you're currently working on?
Yeah, well, so I'll give you the sort of layman's answer to that question because again, I don't have any biology degree. In fact, I've actually um, only picked it up in the last year, and that's part of what I'm trying to show is we think about biology and biotech as these things that exist in these very super high-tech labs where people are wearing white lab coats and you have to have millions of dollars in this space age lab to do this work. But in reality, that's just not true. I mean, genetic engineering sounds like this space age futuristic thing, but the reality is I can pay a company $50 to synthesize DNA, and I can use $15 in reagents to put exogenous DNA into E. coli within my home lab, as an example. So part of the work I'm trying to do is to sort of break down these barriers, these institutional barriers, and also just these mental barriers that society has about who should and who can do genetic engineering and what are those limitations and boundaries. What is biotech? I mean, that's sort of a very broad, open-ended question, but I would describe it as it's a field of technology wherein you're working with living organisms. And so that's very different than computer science, where we're entering code into a non-living machine and it's an input-output sort of machine. With living organisms, we're talking about the fundamentals of life. We're working with DNA, and we're harboring and using these organisms, which have evolved over millions of years to do certain things really, really well. We're harboring them to create food, to create medicine, to solve and heal our illnesses, to produce really valuable compounds. So I'd say biotech is really just a matter of broadly speaking, harnessing nature to bring value to mankind and society and create businesses around that. My particular area of focus, because biotech is very broad and even within specific fields, there's experts in everything. So it's really a field where you're always being challenged and you can never know everything. But because I have an affinity and my start with biology started with growing mushrooms, I decided to make my area of focus in fungi and filamentous fungi. So like I have this petri dish here that's growing a mushroom, um, and fungus is everywhere. It interconnects all plants. Like I can go up pretty much in my yard and capture all sorts of fungus that are living within the roots of the trees, even the grass. It's in the air. If I just put a petri dish out and take the lid off and leave it open for 5 minutes, stuff's gonna land in it and grow. I even have an organism that I cultured from my backyard that once I sequenced it, I found out that it's actually being used commercially to produce a heart statin for people that have high blood pressure. And this is just a fungus that I found in my backyard. So to me, that gets me really excited because we've only discovered a few, just a few of the total fungal species that are estimated to be out there. And so if you think about, there's all these organisms around the world that have been evolving little by little over millions of years to be really good at producing, what's called secondary metabolites. They're able to take the nutrients from their environment and for different biological reasons, they're able to produce new compounds. So a lot of what I'm doing is a lot of exploration. It's going out into the environments, it's capturing fungus, it's identifying them, sequencing them, and studying them a little bit more closely to see like, hey, maybe this fungus has some genes that I can take advantage of. I can maybe take these genes out of this fungus that cause it to produce this medicine, and I can actually put it in something like yeast, which is really good at scaling up and growing lots of that compound in a lab type setting. So that's kind of my area.
Yeah, it's very fascinating to me that we've been using biotech or biological processes of microorganisms, right, for the last 6,000 years, and that has like helped us make so many food products, bread, cheese, preserved dairy products. So to me, that is fascinating how biotech started off with such a basic fundamental principle, right, in the food product area, and that now it's expanded to significantly and just fundamentally change life, and whether it's for clean energy, or reducing our environmental footprint, or what you're doing. It's fascinating how each field kind of is helping to improve life.
Well, we truly do have a symbiotic relationship with fungi. I mean, just to use your example on food, think about alcohol. I mean, yeast is designed to consume sugar, glucose, and excrete alcohol, and humans have taken advantage of that for a very long time. In many cases, I think beer and wine used to be the only things you could drink because there was no access to clean water. We use fungus to now, you can go to a farmer's market and buy fresh mushrooms to eat, which are very healthy for you and a great protein alternative. But we also use them to grow antibiotics. Almost all of our antibiotics come from some sort of fungi that we've discovered in nature that have natural antimicrobial properties to them.
Hey Josh, you mentioned that you pretty much hadn't been in the field of biology since since your whole life except like the past year, and I found that really fascinating. How did you start? Like, did you just like, I'm just gonna study biology, or did you pick up a textbook, or get some online courses? What did you do?
Yeah, I wish I could tell you that it was clean cut part of a plan or a larger strategy, but that's just not the way it shakes out. And that's often the case with life. We have plans, but then life happens. And so really what happened was I sort of got burnt out multiple times in my tech career. I'd been part of lots of startups. Most of them didn't make it, but if you go through the journey of a tech startup, it's quite it takes a toll on you physically, mentally. It's very difficult. And then even in the corporate environment of the tech world today, there are lots of things that are happening culturally within corporate tech, which are just such a turnoff to me and make it very difficult to work in those environments. So I sort of reached this crossroads in my life where I was like, what am I going to do? I've invested my entire life in this one career, and I got depressed. I really struggled with that. And that led me through just a series of events. I had this psychedelic trip, and I came out of that with this incredible new perspective on life. And I simultaneously, I was following a couple of YouTubers and reading some things about, the cost of sequencing going down. You'll hear things, even if you're not in biology. I think most people have heard some things that are on the edge of kind of what's in the atmosphere about biotech, like CRISPR. People, you may not be an expert on CRISPR, but you've probably heard that word thrown around more recently. And so I kept getting these signals from my environment that, hey, there's something here with genetic engineering. I'm hearing more people talk about it. I'm hearing how it's more approachable and you can learn it, that maybe you could do it at home or in a DIY lab. So when I had the psychedelic trip, which came from mushrooms that I grew, I already had some connection to fungi, which is in the biology world. Growing mushrooms isn't the easiest thing. It's not like putting a seed in a plant and watering it. It's a little bit more complex to grow and culture fungus. And so I had that initial connection to biology with that. And then once I, had the psychedelic trip and I kind of realized, I can't go back to the way that I was before in the tech world. I really want to learn genetic engineering and I really want to take my passion for mushrooms to the next level. I just really went deep into, I'm going to learn everything I can about fungus and I'm going to grow a bunch of mushrooms. That was the first thing I did, was like, I talked to people all over the world. You can go to eBay, you can go to Amazon, you can also just network with people online in the mushroom growing community. And I gathered a whole bunch of different cultures of different kinds of mushrooms from all over the world and said, I'm just going to learn how to grow mushrooms. I'm just going to learn how to grow, different mushrooms require different things. And so in the process of learning how to grow mushrooms, you actually learn about the organism itself. Then the next step from there was like, okay, I can grow mushrooms. That's great, but I don't want to become a professional mushroom grower. That's not what I want to do. I want to go to the science. I'm trying to get to the genetic engineering. So the next logical step for me was to start getting into the DNA world. And so I found that there's this thing called DNA barcoding, where you can actually extract DNA from the fungus, sequence a specific area of its genome, and you can compare it with all the other known sequences in the National Genetic Database called GenBank, and you can identify fungus using DNA. And so I learned how to do that. And I learned how to do that from my home environment. And so that allowed me to get DNA, that taught me how I can do DNA extraction and get DNA out of fungus and even get access to sequencing. And I can now do DNA barcoding for less than $7 per species, which is, I don't think people think that of doing DNA work of being so cheap and affordable. But, in a couple of hours, I can do DNA extraction from fungus. I can mail it off to a lab, and within a day or two, get a sequence back and have access to that data and share it with the world. I can do it for less than $10. And that's pretty incredible. And so the next step after that was like, okay, I can get DNA out of fungus. Now, how do I get DNA in it? And that's the big thing that I'm working on now, and that's called genetic transformation. So that's, how do I take genes from other organisms or other fungi, put it in a fungus or another organism, and get it to express those genes? How do I do that? So, it's a long story, but basically the idea was I started from the very beginning. I had an attachment with fungus from growing the mushrooms, from having the psychedelic experience that sort of grounded me and connected me to the nature aspect of it. And then because I've spent so many years in a call it an intellectual field, computer science is, anybody can type on a keyboard, but it's really a job of thinking. You're really thinking about architecture, you're thinking about design, you're thinking about how systems interact and operate with each other, efficiency. So I just took that same part of my brain, which was attuned to code, and I just made the connection to DNA. And when you get into genetic work, you realize there's actually a lot of similarities between writing software and writing DNA. It's a different language, but all genes are encoded with a series of nucleotides. So you can code for genes. And there are some basic tools, molecular biology tools that you can use to get exogenous DNA into the fungus. And once you realize what those are, all of these ideas that you have about how difficult it is or how expensive it is, you start to go like, wow, this is really incredible. And why are they keeping this a secret? It almost feels like, the information lives in these papers, academic papers, which is sort of the currency for grad students. And then when you look at the field of biotechnology, and you look at the startups who are starting biotech companies, they're all PhDs by and large. They're all PhDs, or at a minimum, they're grad students. But why is that? Why are there all these like barriers that are preventing the average person from getting access to the knowledge of how to do genetic engineering? Although I'm doing genetic engineering for fungus, actually the process is very similar, whether you want to genetically modify humans or you want to genetically modify plants. The same underlying tools and technologies are very similar, and they're not that difficult. They just happen to be behind a paywall in a paper. And from a societal standpoint, most of the money is being given to people who follow a particular path. They go to grad school, they graduate with their PhD, and that opens up opportunities for them. But I don't think we want to live in a world when sequencing and genetic engineering, the cost and the tools are accessible to someone like me who has a home lab in my home, in my kitchen. I don't think that we want to have only this very tiny portion of the population have access to that technology and making decisions for everybody else. I think at a minimum, we need to make sure that we're making this technology understood and accessible to everybody, regardless of their academic background, regardless of whatever background they come from. We need to at least make the information available to them so that they can advocate for the right policies, for the right way of using it. And so that's part of what my mission here of starting Everyman Bio is all about. It's really about, let me do a deep exploration into something like genetic engineering, which sounds space agey and very difficult to do. Let me actually change people's perceptions and show them that it's not as difficult as you think. It's not as expensive as you think, but it's incredibly powerful. And what if we're actually able to open this technology up to the general public? What sorts of amazing things is your neighbor going to come up with? Obviously, there are ethical and there's risk associated with that, but we should be having that conversation. We should be defining and debating what those limits are, and we should be doing everything we can to democratize the information, so at least people understand how it works, so that they can advocate for themselves in this process.
Right, Josh. There's a lot to break down there, and I definitely want to get into genomics and its applications. But just to kind of backtrack a little bit, you mentioned the psychedelic experience. It was kind of like a catalyst in your life, kind of changed you. Could you elaborate on what motivated you to do the psychedelics? And obviously you don't have to talk about it because I know it's a lot personal to a lot of people, but kind of your motivations to do psychedelics in the first place.
Well, I talk about this on my podcast. I interview scientists and other people from the psychedelic communities, and I'm quite open about it. I don't mind talking about it at all. Frankly, well, I should say I come from the South. I came from the South. I came from Texas. I grew up there, and I came out to LA when I was 18, 19 years old. So inside of me, I have this person that is very like straight and narrow. I don't really have a lot of experience with doing drugs. So if you were to ask me at different points in my life what I thought about psychedelics, I would have been very turned off by it, because I would have put it in the classification of, I don't do drugs. I stay away from those things. But as life happens, you evolve and you get new experiences. And when I was kind of going through an internal crisis, I think everybody deals with depression at different points in their lives. I reached a point in my depression where nothing was working. I was in therapy for years, and then suddenly, right around when COVID started to happen, my therapist decided to move out of state. So I lost I lost my number one confidant, person that I used to lean on, and you know, when I was going through a tough time. So I lost my therapist, who I developed a bond with over many years. I had an identity crisis because I didn't want to go back to doing what I was doing for my career. And then, you know, I'm older than you guys, so it's much more difficult when you're 15 years into your career to kind of go, well, I'm going to start over. That's really hard to do. I didn't want to go to school and start completely from scratch. And I had no clear pathway forward. And I was in this sort of philosophical quandary of, it's very difficult to be in a place where you feel like you're stuck. You're stuck in this really negative space. You cannot envision any way out of it. And so, you know, I've tried antidepressants. They don't really work for me. They also kind of depress your ability to feel anything, including happiness. So I tried everything. I was in a state of sort of desperation, but I had been reading about the positive studies that were coming about from people who had psychedelic experiences. And so I had reached a point to where I was like, I was desperate to try anything that might help me. And so my wife, who had actually tried them before, was like, you know what, you grew mushrooms 10 years ago. Why don't you grow these mushrooms, and why don't you have an experience and see if that helps you? So I did. I grew some mushrooms. She babysat me and helped me facilitate a positive experience. And yeah, man, it was eye-opening. You know, like, we can talk. I'm happy to tell you more about the experience itself, but the main takeaway with psychedelic experiences is we don't realize how much we get locked into a particular set of consciousness, a state of consciousness that we all carry with us in our day-to-day lives. And that can become very rigid. And that rigidity often can prevent you from imagining alternate paths or alternate ways of thinking. With a psychedelic experience, it sort of does something in the brain that unlocks, it takes you out of that locked, rigid framework that you're in, and gives you a new way of experiencing human consciousness. And you come out of that with a completely shifted viewpoint of how you see the world. Because whereas before I did the psychedelics, I was in this very depressed, I'm stuck, I don't think I was beginning to feel hopeless. How am I ever going to get out of this position? I have no energy, because depression just zaps you of energy. I have no way of getting out of that. The psychedelic experience forces you to perceive the world in a different way, and you come out of it going, well, I know that there's another way of thinking about this because I experienced it in the trip. So I actually know, I don't know what the future holds for me, but I know that it's not this locked in rigid viewpoint that I had before. I know that there's other opportunities. So that's also what gave me the comfort to kind of say, you know what, I'm going to go into genetic engineering. I'm going to teach myself it. I'm going to share that journey with the world, and hopefully encourage other people to get connected and motivated with genetic engineering. And I don't know where it's going to end up, but I'm going to embrace it as the journey that it truly is, and trust that if I stay committed to it, something incredible like being a guest on your podcast will open up for me. And that would not have been possible without having had that psychedelic trip, because I was too much in a place where I needed to have all the pieces figured out. I needed to have a very clear-cut plan to a destination. The psychedelic experience gave me the flexibility to kind of go with the flow a little bit more. Just know that you're on a journey. You're not going to have it figured out. You're going to make mistakes along the way, but it's going to be okay. It's okay to not know the destination for the first time in your life. It's okay to actually just to see what happens.
Gotcha, Josh. That's honestly, I've never done psychedelics myself. I'm not a drug person myself, but that's very interesting from like the viewpoint where I'm at and you were to where you are now. So it's quite interesting. We talked about biotech and kind of like the basis of it. Can we move into genomics and like how genomics varies from biotech and what the real difference is?
They're two very much related. You know, in the early days of biology, we didn't have access to cheap sequencing. We didn't have access to the data that, you know, what's incredible is at the heart of every organism that we know of on planet Earth. And as far as we know, we don't have any evidence of anything different in the universe, that all life is fundamentally DNA based. That means somewhere deep into into the cells, there is a strand of nucleic acids, this DNA molecule, which is this incredible molecule. So whether it's fungus or a plant or a microbe or a human being, fundamentally, we all have this incredible molecule that's the same within us. The only difference is the individual nucleotides and the order that they're in. So genomics is really a matter of being able to sequence those long nucleotide sequences from the DNA molecule and to be able to analyze them at scale. So I forget how big the human genome is, but I can tell you many of the fungal genomes, if you were to sequence the whole thing, they'd be about 40 million base pairs long. So if you think about what is a base pair, and it's represented by one of the letters A, T, G, or C, imagine 40 million of those. And you have that in a database somewhere, and you can compare that between different fungal species. Well, genomics has allowed us to kind of look at those very large data sets of sequencing data and kind of find within those sequences, where's a gene that produces, this cool protein, there's different transcription factors and different things in the DNA that we can actually find by exploring and doing analysis on that genetic data. So genomics is very much interrelated with biotech. In fact, it's moving more into the software and data analytics side because the cost of sequencing has gotten so low. It's very cheap now to go out and collect a lot of different organisms, to sequence them, to collect that data, put it in your data warehouse, run all sorts of analysis on it to look for new and interesting genes or molecular tools that we could take. And once we've identified a gene from an organism that produces, say, here's a gene that produces a protein that has antibiotic characteristics, we're going to take that gene out and we can actually put that gene in other organisms that are really good at expressing that gene. So we can actually move genes around between organisms and leverage what each individual organism has evolved to be really good at. For example, we talk about yeast. Yeast is used widely in biotech because it's very good at fermenting at large scale. It's very good at, you feed it sugar and it's good at producing alcohol, but it turns out you can put other genes for producing medicine in the yeast, feed it sugar, and it will excrete the medicine at scale for very cheap. So that's really genomics and synthetic biology and genetic engineering are very tightly coupled. And right now there is a massive rush to sequence as many things as possible and to do the analytics to make new discoveries from that data.
Yeah, Josh, as someone who's never studied genomics and biology, could you explain the process of gene editing? And could you also, kind of a follow-up to that, explain the technology that's used? So, you mentioned CRISPR before and other technologies. Could you also elaborate on that?
Yeah. So the process of gene editing starts with you designing what DNA you want to go into the organism. And so there's a way of actually finding genes. So I'll give you an example. There's a gene that comes from a jellyfish that's called the green fluorescent protein, GFP. And this gene was discovered not too long ago, and the scientists who discovered it and isolated it from this jellyfish, you probably have seen bioluminescent jellyfish, at least seen pictures of it. Well, actually, those jellyfish, they have this gene that produces this protein that happens to be fluorescent under UV light. And so they isolated that gene, which again is just a string of letters or a string of nucleotides. They were able to extract that gene out of that organism and put it into other organisms. So the gene editing process starts with identifying the genes, identifying a couple of the machinery that you need to tell the host organism to process that gene. If I just put a gene inside of an organism, it may not recognize that gene and it may not do anything with it. What I also need is a promoter, which tells the machinery of the organism to process that gene and actually produce the protein or express the protein that I'm trying to get it to express. And then a terminator, which is at the end of the gene, which tells it to stop producing it. So gene editing is really, the first step is really putting together your payload. And I do this because often what that payload looks like, it's called a plasmid. If you do a search, anyone listening, do a search for a DNA plasmid, you'll see these circular images that are representations of genes and all of the machinery that's necessary for the host organism to actually express the genes in the plasmid. The next step is actually getting that DNA into the organism. So once I've kind of designed the DNA, I can actually hire a company to synthesize it and send it to me in a little vial. They'll send you a vial of the DNA, the genes that you kind of design. They'll send it to you for about 12 cents per base pair to give you a sense of cost. They'll synthesize it in a lab. They'll send you the DNA. And now the question is like, okay, I have this DNA in this little vial. How do I get it into the fungal cells, like some that I have growing here? Well, for fungus, there's a lot of different techniques you can use. The technique I'm using is actually using something called agrobacteria. So what I'll actually do is I'll put that DNA into this special bacteria, and then I'll put this bacteria with the fungal cells. And this special bacteria has the ability to transfer some DNA to whatever organism it's in contact with. It almost infects it. So I will literally mix some of the agrobacteria with the fungus together, and it will transfer the DNA that I put into the bacteria. It will transfer some of that to the fungal cells for me. And then I just grow it out, and there's a few more steps, but that I'm using nature, I'm harnessing nature's own ability to do gene transfer to get DNA into the fungal cells. There's other less complicated ways. For example, I have a machine sitting behind me right here. This is called an electroporator, and all it does is it's a glorified shocking machine. I have it's a little plastic container called a cuvette that has 2 electrodes on each side. I could put human cells in there, and I could put some DNA plasmid in there, and then I configure the machine I set the voltage and a couple of other electrical settings, and it will zap it. It'll send a very high voltage, very fast, high voltage pulse through it. And what will happen is that DNA will actually go into the human cells, or whatever cells, could be fungal cells, could be bacterial cells. The DNA will enter into the cell will recognize the DNA, and it will start processing it like it does any other DNA. And that's pretty simplistic if you think about it. It's using a high voltage shock to shock the DNA into the cells. That's used today in labs to do CRISPR experiments. CRISPR, they'll design a DNA plasmid, which has the CRISPR protein in it, or Cas9 protein, and their guide DNA, basically just their DNA, and they'll mix it with human cells, and they'll shock it, and the DNA will go it'll go into some of the human cells, and then that's it. They'll let the human cells grow, and some of those human cells are going to take up that DNA and start processing it and express the genes that were put into it. And in some cases with CRISPR, it might be that it actually cut some of the DNA out of it, or deactivated a gene, or it inserted a gene in a particular location. And electroporation still is one of those things in biotech that no one really knows how it works, which is kind of interesting. There's a lot of theories about how this machine works, but it just is a staple of one of many ways of doing genetic engineering. And yeah, so there's many different ways of doing it. You can shock the DNA into the cells. You can treat the cells where you basically put some chemicals in there, and it loosens up this membrane, or it like degrades the cell membrane, so it's easier for the DNA to flow through the cell walls into the organism. Or you can use agrobacteria, and you can harness nature's natural ability to transfect genes into an organism.
To me, the most fascinating part of genomics is CRISPR, right? This new gene editing tool. At the basis of it, I mean, from my understanding, CRISPR arrays, right, basically allow bacteria to remember viruses, and that's can be really helpful in things like cancer research, where we know scientists have discovered, right, that changes in DNA essentially cause cancer, and we can use CRISPR as a gene editing tool to, help remedy that. And the cost of these technologies, I mean, you mentioned you're doing gene editing in your lab, and the cost of this has significantly gone down since 2003, 2004. For me, I'm really fascinated in the investing part of genomics, right? I don't know if you're familiar with Kathy Wood. She's, obviously a very renowned hedge fund manager of ARK Invest, but she believes that genomics, and I agree with her, is going to completely change the world in the coming next 5, 10 years. I mean, this is not something long term. I think it's already here and it's starting soon. And so, in the business aspect of it, earlier you mentioned that most startups fail, like most of them, like overwhelming majority fail. In this new field of genomics, you mentioned that a lot of, in biotech in general, a lot of people who are starting these companies are PhDs, right? Who don't have maybe a business background. They understand the technology and the nuances of it better, but because they don't have this financial understanding, maybe that's the reason why they can't get off the ground. In your mind, one, what do you see as the business model of genomic companies, and how can startups not make some of the same mistakes that previous startups in the space have made?
Yeah, well, there's some things about what you asked that I'm not the expert in, so I'll talk generally kind of about just business and approaching startups, but also just about the market economics of biotech. If you have this technology, biotech and genomics, which is rapidly decreasing in its costs and the barriers to entry are being lowered, but the value that you get from it is going through the roof, then that's going to create a large demand for new biotech companies and new genomic companies. And accordingly, just like we saw in computer science over the last decade, what's going to happen is that's going to drive a large demand for the workforce. And so we are rapidly going to be approaching a phase, in my opinion, where you can't just say we're only going to fund startups that are founded by PhDs and masters. There's just not enough people to create all the companies that could exist out there. And so I think what we're going to see is a shift to people that come from a variety of different backgrounds, bringing their expertise and knowledge from other industries into biotech. Genomics, for example, is a very computer science heavy field. There aren't a lot of differences from a pure software standpoint and a computer science standpoint. There aren't a lot of differences between working with large-scale genomic data and large-scale credit data or large-scale any kind of data. It just happens to be the underlying data is genomic data, not financial credit data, as an example. And so already we're starting to see a big overlap in the demand and the need for computer science skills and expertise in the field of genomics. All the same rules though for startups still apply. You still need to do the work of validating your idea, of trying to be very close to your potential customers and understanding their needs so that you're developing your product so that there's a market demand for it. I would say though, one of the opportunities and part of what drives the work that I'm doing is that people are really naturally intuitive and creative and very good at solving problems at scale. If we could get people who, like myself, I don't have any biology degree, I don't come from a background in biology, but in the last year, I've taught myself genetic engineering. If we could get more people engaged at some level, imagine the type of ideas that would come out of that. Imagine if we have someone who comes from the food industry and maybe they have this idea about genetically modifying yeast to produce something incredible that maybe mainstream institutions and scientists kind of aren't looking at, they're not thinking about because they don't come from that perspective. They don't have that viewpoint that these particular people do. That's where I think the potential opportunity comes from, is being able to take the academic folks and the institutional players and partner with people like myself who are sort of the enthusiasts, the people who are looking at organisms from a different perspective and looking in places that maybe the big players don't have an incentive to look at because it's maybe too small for them. But it's very possible that I could go out, for example, collect a fungal species that is producing a new antibiotic that is incredibly valuable. You know, we have an antibiotic shortage right now in the world, and because, you know, nature is always evolving, and any new antibiotics we introduce, as soon as we introduce them to the environment as a medicine, the environment is going to respond back by developing immunity to those antibiotics. So there's always this cat and mouse game. But if people knew that you could potentially discover a new antibiotic just by going in your yard and collecting some fungus and learning how to isolate it, that's really powerful. It's almost, you're almost crowdsourcing molecule discovery, antibiotic discovery, pharmaceutical discovery. So I think, to kind of go back to the business aspect of it, there's going to be a need for increased people in the job force, especially people that have computer science backgrounds, but also, as you mentioned, people with business backgrounds. If you went and did a PhD, you know, and you're now starting a company, chances are you spent most of your time on the science side, not on the entrepreneurial side with how do I talk to investors? How do I find a customer? How do I even take an idea that I've incubated in the lab and scale it up to a business that could serve millions of potential customers? Those are all skills that require different side of the brain than someone who's been in a PhD learning molecular biology. I am hearing, though, from people who are in the final, you know, who are in their PhD programs now, that we're starting to see more schools incorporate the entrepreneurial training and startups within their curriculum. You know, in my experience of business and working with startups, nothing beats the hands-on experience of actually going through the process of joining a startup. You know, when you join a startup, you're not doing one thing. You're going to be doing 100 things. You'll probably also be the same person cleaning the bathrooms at the same time, because who else is going to do it? So you're doing a lot of different things. And so whenever you get into that realm, it's more about mindset and less about actual skills that you went to school for. It's more about embracing the idea that, like, you're on a learning journey. You're going to be exposed to a lot of different things. You kind of need to develop a lot of different skills to be a good startup founder. You have to know the science, but you also need to know the business. You have to have good, you know, interpersonal relationship skills, the ability to build relationships with customers, with investors. There's a lot of different things you need to know. So for people who are, you know, wanting to get into biotech, yeah, the same advice I would give to anybody who's interested in starting a tech startup is join another startup, do it for a year. And I mean, the earlier, the better. It's a very highly risky venture, but you're going to learn more in that 6 months or a year than you probably got in your entire PhD, just from the hands-on experience of, you know, there's a lot on the line when you have a startup. You have potentially have employees, you potentially have investor money, and you have your reputation. You really want this thing to succeed, and so there's no other experience that beats the real world experience of joining a startup and going through that journey.
You know, to me, the world of technology, it's fascinating how the democratization of things has replicated into different fields of technology, specifically for genomics. And I think, as someone who, you know, who follows this space, the investing side of the space, it makes me optimistic, you know, seeing that you yourself have started to, you know, do gene editing in your own lab, and that's just going to continue to accelerate. And I think the business community of Wall Street should really catch up to that, because I mean, besides Kathy Wood, who manages the hedge fund ARK Invest, you know, she invests in disruptive innovation, and she has an ETF based solely for the genomics field. I feel like a lot of Wall Street firms are catching up to investing in disruptive innovation, specifically in the genomics field.
Yeah, I agree with that. I will play devil's advocate, though, and say that if I'm an investor and I have someone that says they genetically modified an organism in their home and now they want to start a business, that would seem a little bit riskier to me, because you're dealing with something that is a little bit outside of the norms of what I'm used to investing in. You know, because I don't have those letters attached to my name, because I didn't go through formalized training, I'm self-taught. Now, there are pros and cons to that. If someone approached me and were like, look, I spent the last year self-teaching myself genetic engineering, and here's irrefutable evidence that I was successful at doing it in my home lab, that would mean a lot to me. I personally would have appreciation for that, because that's the journey that I'm on. But I can also understand how the investment community, the business community, who are accustomed to working with PhDs that follow this very rigid pathway that's sort of been created for them to create businesses, I could see how that would be a little scary. It's uncharted territory. So I think, you know, with anything where you're disrupting, the first few people to do it always have to overcome these really big challenges. And so that's what I'm dedicated to doing, is kind of showing that it can be done. And then just like the people before me who kind of encouraged me, just the various people on YouTube and people in the biotech community who showed me that there were things you could do from home that I didn't know were possible, I'm hoping that my journey will inspire others as well to kind of go, well, if he did it with fungus, I could do it with plants, or I could also do it with fungus and take what he did to the next level. You know, I'm showing that those boundaries can be broken, and hopefully that creates an avalanche. In a perfect world, what I would love to see is more startups where there's a blending of startup founders, some that have the PhD, some that don't, and have those startups leverage each other's strengths, because that's where you get some really exciting backgrounds. You have a real estate, you know, Matt has a real estate background. Who knows what sort of ideas he's going to have from that perspective? I can make connections immediately between real estate and biology. And him bringing that into a biotech, it might seem at surface to be completely not related, but actually he's going to have a lot of ideas that he's formed from his own life experiences that will inform how that business is run. In fact, he might be the great COO, Chief Operating Officer, or the person that manages the investments, or the person that talks to potential pharmaceutical companies who want to leverage this technology, because there's a lot of interpersonal interaction in real estate, and not a lot of analysis. He's on commercial side, so he's doing a lot of underwriting of deals. So there's a lot of overlap between those particular skills and the skills necessary to run a business. So I think what we'll see, and I think what a savvy investor will begin to notice is, you know what, I'm not looking for just to invest in a purely scientific endeavor where all the founders are just pure scientists. There's actually a lot of risk baked into there. Yes, they have the PhD. There's no doubt that they're smart, but they don't necessarily know business. They don't necessarily know how to scale that business. Do they know how to hire people? Are they only going to be myopic in their point of view in terms of only hiring other scientists? What about, what about a self-taught computer science, self-taught software engineer who's an amazing coder who would love to get hands-on with genomics? Are they going to pass over that person because they don't have a PhD? So I think what we'll see, and I think what investors will tap into, is like, they want to see the whole picture. They want to see a well-rounded company that addresses a lot of the risk areas in starting a new business. And that's where I see a lot of potential in the business arena.
Yeah, to me, it seems like just computers all over again. Like, in the early days, computers were only accessible to like PhD, and more academic folks. And then, now we can make a website at your home, just sitting at home. And it seems like that same trend in biotech.
Absolutely, absolutely. And what's funny is, and I'm glad you caught on that, I've talked to a lot of people who go, no, it's not the same. Biology is so much less predictable. It's not as discrete as computer science. And there's some truth to that. When you're working with organisms, there are things about them that are very difficult to understand and not predictable. And you can run an experiment, and it's not like computer science where it's like, it does exactly what you code it to do. There are things about biology where you tell it to do something and you get a completely unexpected result.
It's unpredictable, yeah.
It's unpredictable. That said, we are moving into a field and an area where the predictability is rapidly going up. Because the more that we study these organisms, the more that we do genomics and we understand the nature of DNA, the more we're able to understand exactly how it works, and we're able to get it to do the things that we want it to do. So absolutely, I think we are in the early days of it getting to a point where computer science was, where you needed to have a highly specialized skills and training. You needed to have a lot of money because computers were expensive. Now, the cost of sequencing has gotten incredibly cheap, and the computing already is super, super cheap. And we're even seeing the actual hands-on biology, I'm noticing, is even becoming automated, in the sense that nowadays you can design an experiment on a computer using genomics. You can have a software engineer design all of these biological genetic engineering experiments, and they can use an API to send it to a company in the cloud that will use robots and run the experiment on the organisms remotely. And now I don't even need to have a lab. I can just, from my house, write some code, design my genetic experiments, choose the organism, choose programmatically how the experiment should be run, send all that through this API. This company that has a highly automated robotic lab will run my experiment, will take all the data, will publish it back through the API. Now I'm just sitting at home running these biological experiments purely from a computer. That's kind of where we're headed in biology.
So I like that statement. As predictability goes up, it's more and more safe to do it, right? We take a look at like GMOs, and that's a big thing going on. And from my understanding, is people don't really understand that it's actually a good thing. Like if they took a look at a banana before GMOs hit it, they wouldn't have eaten that. If they took a look at corn before GMOs hit it, they definitely wouldn't have eaten that. And so my question to you is, like, as time goes on and like we chug along and we take a look like at Dolly, the sheep, and I believe Zong Zong and Hua, the two monkeys in China, how far do we go? Like, where is the line cut? Where do you think that ethical line should be placed?
Well, I'll start with the first part of your question. That is a fantastic question and something that I think about as well. So the first part of that question is, people have a natural fear of the word genetic and consuming things that have been genetically modified. But that fear doesn't always come from, fear is an emotion. It's not a rational thought. It comes from a different part of the brain, which is feelings, as opposed to rationality. So what we have to do to overcome that inherent fear, like with anything, is we have to educate people, because people don't know what genetic modification is. And so if you don't know what it is, and you know that it's very powerful, and you know that it could have big implications on your health, then yeah, you should fear it. But we can turn that fear into a respect. And we do that with knowledge. And so, but what people don't understand is, we've been genetically modifying organisms since the beginning of mankind. We do it through selective breeding. We do that by going, this piece of corn grew bigger and faster than this one, so I'm going to breed that one more than this one. That over time creates genetic modification to this corn instead of this one. What we're now talking about is, we're going to not go through selective breeding, but we're going to use molecular tools in a lab to do that, to speed up the pace of genetic modification. And so just because you're doing it a little bit differently doesn't mean it's worse. The question is, what is the genetic modification that you're making? That should be the question that people are asking, to decide whether or not they should be fearful of it, if they should put a halt to it, or if they should embrace it. Because yeah, you can GMO something that's incredibly dangerous and pathogenic and could kill someone. Or you could genetically modify a potato to last drought conditions and prevent famine. So those are two very different use cases, but one of them is saved millions of people's lives over a millennia, and the other one could be used as a weapon. So hopefully that answers the first part of the question around fear. We have to educate people and get people, asking, what does it mean for something to be genetically modified? Just because something is GMO doesn't mean it's inherently negative or bad. The second part of your question was around, how do we define what those limits are? And that is a great question. So I will say, I don't know the answer to that. I don't think a lot of people do, because we're in uncharted territory. I am showing, there are people who are naturally apprehensive and fearful of what I'm doing. I am doing genetic manipulation of an organism in my lab under no supervision. I'm accountable to no one. Could I engineer this fungus to be pathogenic to humans and maybe cause disease in humans? And then all I have to do with this fungus is release it out into the wild, put it near a plant or put it somewhere where it can grow, and it will grow and spread. And now I've introduced a GMO fungus with unintended consequences out into the environment. That is a possibility. That is a possibility. And I think we should have some limits around that. Even though I'm doing it and I'm not planning to do any of those things, not everybody's me. And you guys don't know me. The general public doesn't know me. How do you know that I don't have bad intentions? So we need to have some guardrails. I'm not advocating that we be the wild, wild west and people do whatever it is that they want to do. That said, I don't work with anything human, not because I can't. I could. I could be running CRISPR experiments here, which is pretty cool. And I would love to show people to do that. But I think it's too controversial. And my intention is not to push the ethical boundaries and to try to break any ethical boundaries or push them or to make people feel uncomfortable. I have a completely different set of goals and aims. I'm trying to do things safely. But the question still stands. Could I? Yes, I could. In fact, there probably are people right now who are CRISPRing human cells to do all kinds of things, maybe solve diseases, or maybe as the technology becomes more abstracted and easier to use, and there becomes the cost of entry is lower, you're going to have someone that just wants to see the world burn, right? You're always going to have people out there who are like, oh, I have this incredible power. Look how easy it is for me to put this viral gene into the human genome and infect people. Let me try it because there's some kind of like power that they get from it. Yes, I mean that's a real concern. I will say though, like where the technology is at is not so easy yet that you're going to have. Someone has to be very motivated to learn all the things and to do that. So if it was going to happen, it's more likely it's going to happen from an existing lab or someone who's in a lab that has that knowledge already and access to the tools to do it, which we are all living in a world where that may be a reality with COVID. Maybe we don't know yet. That's something to be explored. Go back to the basics. I kind of believe in the golden rule. I wouldn't want to work on anything that I wouldn't want to be exposed to or I wouldn't want other people to be exposed to. But it's a really good question is what are those ethical and philosophical boundaries to set? And what laws should we construct to keep it safe, but to not slow down innovation? Because there are other countries in this world that have none of those ethical concerns that we in the United States are constantly debating and philosophizing about. Meanwhile, there are other actors in the world that are moving forward, full steam ahead, putting very little concern about the long-term implications of the research that they're doing and the potential harm that they may do to the globe. And the only way we're going to be able to counter that is not to put our heads in the sand and say, no one should be doing this research. We have to be doing this research. We have to be defining what that line is so that we can hold other people accountable. And we should be doing it in a way that's smart, that allows us to continue to innovate, but keeps it safe. And so there's always going to be a tension between making it, it's like regulation in general. It's not bad in and of itself. It's meant to protect us and it's meant to protect one another. Too much regulation, though, can stifle business, can stifle innovation, and can slow it down. So we're always in a tension in life and in society between how much constraint do we put around something to keep it safe and to protect people, and how much do we allow the freedom to explore and to innovate and to experiment. And there's trade-offs with both sides. So we're going to constantly be in a tension between those two worlds. And that's just life. We have to work through those, but we can't ignore it. And I think we're not quite at a point where we're giving it enough attention, and we're not having enough of the conversations and debates around the potential side effects and harms of not allowing or allowing genetic modification. It could very well be in the next 5 years, once more people like myself are publishing their research to the public and showing how easy it is to genetically modify organisms in your home. It could be that that's illegal in the next 3 years. I hope it's not. I hope it's not, but it could be. And we'll see. That's part of the journey here is with any new technology. You know, when Uber and Lyft first started, they were operating outside the legal boundaries. That had never been done before. We had only ever had licensed cab drivers that were driving people around. And so sometimes when you're innovating or disrupting, you are entering into a gray area legally because it's unexplored territory. We never had to deal with an Uber or a Lyft type situation before, so we had no case law for it. We had no philosophy. We had no, you know, we had to figure all of those things out as we go. That's just the nature of technological advancement, though.
Right. That is very, like, it's very interesting because we know, we go on, and just as you said, this is not a linear path. You know, different nations are doing different things. They have different ethical boundaries regarding, genomics and how we play about this. So how would you say, how do we correlate things? Because obviously countries don't care what another country does, and some countries do care what another country does, right? And it goes hand in hand. And, say as an example, just say the US, we're very stringent about these things, right? But China, on the other hand, is not. And so how do we go ahead and communicate with one another and, try to draw that connection so that scientists are on that one-to-one basis?
Yeah, there is something special about science and history where scientists have transcended political and international boundaries to cooperate with one another in the interest of scientific endeavors. And I love that, right? Because anything that interconnects us as people, science and knowledge is always one of those things will transcend our differences, our cultural differences, because we're both just trying to understand the world that we live in. And we saw this with Einstein interacting with scientists across seas to try to stop nuclear proliferation, for example. So I would like to see more international collaboration with folks in China. And I think it does happen. It definitely does happen. We also have to trust but verify. China is a country with a proven track record of keeping secrets. Russia is also one of those countries. And the US has secrets too. We're all keeping certain things away from each other, and we have competing interests in that regard. And there are certain times in history where the tension is going to be so high, it's not enough, it's too much to overcome for two independent scientists to work behind the scenes. So, I think the best thing that we can do as Americans in America is to invest in our own research, and so that we are not only innovating and creating new medicines and new technologies here, but we need to be able to protect ourselves should we have a bad actor deploy something on our country as well. So we need to be investing and advancing our own sciences forward, but always extending an olive branch for collaboration with our fellow humans that are whatever part of the world. They're also scientifically endeavor, that are pursuing scientific endeavors, but we have to do it intelligently. This is one of those things, when you talk about genetic engineering, you're talking about the mechanics of life. If you make a mistake and you say design the wrong virus for whatever reason, and there are some valid reasons for studying viruses and doing what's called gain of research. You might have heard that in recent vernacular, political vernacular, but there are valid reasons to do gain of function research. We need to know what makes a virus deadly to human beings. We need to study that because we need to be able to create a cure for it. But that has a lot of risk, very highly elevated risk associated with doing that kind of study. How much of that collaboration on that particular project should we be doing with another country that may not have the same interest, that may have an interest in actually undermining us for whatever reason? So those are the questions we should be answering. We should be very careful about that. So I don't think, it's like most things in life, there's no black and white answer to this. It's nuanced. It's very individualized on a case-by-case basis. We need to be paying attention to it. We need to have checks and balances in place. We should be encouraging collaboration. That's always much better than isolating ourselves off from other countries and other people. But we should also be mindful of, they are their own country. They are loyal to that country and that culture, just like we are. We should protect ourselves, just as I'm sure that they are as well. It's a very nuanced way. I don't think there's a blanket way that we'll be able to solve these very challenging predicaments, especially when you talk about working with biology or potentially biological weapons, and even things that may not be intentionally biological weapons. There are a number of cases where a genetically modified organism has been released into the wild with negative consequences. And it was, not intentional necessarily, but it happened. So it's very powerful. And I think that's probably why so many people have a natural instinct towards fear, because it is very, very powerful. If you can master the molecule that controls all of life, well, that's a whole level of mastery over life than we've ever had in history before. So that's pretty incredible.
I think with any technology, there's positives and there's a downside to it. In the 1990s, Jenish was talking about how computers weren't accessible to a lot of people, and we all thought they were just not accessible. And in the beginning phases of the digital revolution, it was really positive, and we didn't really see the spread of disinformation that now happens on social media, and the downside of what the internet can produce. And similar with genomics, I think there's a positive side to it in terms of helping people cure diseases, honestly, and improve lives on a health level. But on the downside, countries can use this as a way of use biotech as a way of terrorism, right? Releasing pathogens, creating or rather forcing onto pandemics and using that as a way of biowarfare. I think that's a possibility as countries have different, priorities.
Or it could be an accident and they just don't want to tell anyone because they don't want it to look bad on the country as well. Well, that's equally as dangerous because the end result is the same.
I agree. And since you brought up this point, I want to ask, and I think you can provide a valuable perspective on this. COVID, it was obviously, one of the most consequential things that ever happened in the world since the 1920 pandemic. Now there's a new theory that's out there. In the beginning, this started off as a conspiracy theory, right? That this was a lab leak. This was created in Wuhan. And now, there's been a lot of good journalism made, and now we have more information. And so I want to provide some context. So the Wuhan Institute of Virology has been studying, coronaviruses for years, because that is really endemic to that region, that coronaviruses. And there's a theory that perhaps researchers there were studying this and accidentally it leaked out and China tried to cover it up. Since you worked in a lab before, what are the chances that this has some veracity to it? Certainly, all the politics aside, I think it's possible that this happened. It's very much possible. I mean, we can't really trust what China plans on doing, in my opinion. But if you want to provide some perspective on this, what's your
opinion? No, I love it. Hopefully it doesn't get you banned from YouTube to even have the conversation. Your vaccine hesitancy score is rising. No, unfortunately, this is one of those topics that used to not be politicized. This isn't the first pandemic we've had. We've had other pandemics. We had SARS during the Obama administration. It wasn't nearly as politicized, but there were a lot of things in American culture in the last 4 years that have led to a highly polarized society. That's not a shock to anybody. I mean, everybody knows that. And so everything became politicized. And that's a danger to all of us, regardless of what side you're on. For everything to be viewed through the viewpoint of politics, incredibly dangerous, because you lose objectivity. You lose the ability to just look at the data in front of you and to trust it. You can't do that anymore. It's actually very, very difficult to discern fact from fiction on anything COVID today, because there are so many competing interests around the board. I can find 10 studies that say masks work. I can find 10 studies that say they don't work. I can find emails from Dr. Fauci that say they don't work and some that say they do work. So what's true and what's fiction? Very, very difficult these days. We are inundated with information. That said, the information that I've seen about the lab leak theory, it's all circumstantial. We don't have anything definitive yet. So in the court of law, that wouldn't go as far as something definitive. But I do believe in Occam's razor. The simplest explanation is usually the best. We have a lab in China, in Wuhan, that was studying novel coronaviruses. We have the first 2 people that got infected were from that lab in November. There are a lot of things about the circumstances that point to it was probably a lab leak. I could be wrong about that. We need to do that research and we need to figure out the truth. We've not found a host animal yet. That's not entirely odd. We haven't found a host animal for a lot of pandemics. So that, use that information for what you will. Is it a possibility? Yes. Do I think that there's more circumstantial evidence pointing towards a lab leak? Viruses are incredibly small. They're very, very small. They're a little bit bigger than DNA. It's very easy for those things to hop on a surface and get outside of a lab environment. So I think it's entirely possible. I don't know that I see anything that says it was intentional, but I do think it's possible that because China doesn't have as strict, they don't have the shared values and ethics, and we operate independently, we operate differently. I do think it's possible that it leaked. I do think it's possible that they had an incentive to not share that with the world. For you can imagine how embarrassing that would be, how poorly that would reflect on the scientific community. I don't know if that answers your question. I absolutely think it's possible. I can't say one way or the other. I definitely think we should know, and I definitely think we should research it. Screw the politics. We need to make sure something like this doesn't happen again. And if it turns out that we did fund research, which it looks like the NIH did fund some of the research going into gain of function research in the Wuhan lab, well, we should know that, and so that the American citizens can decide where we want to invest that money in the future. We have 11 labs, I think, in the United States doing similar research. Maybe for a period of time, we need to redirect those funds back to American labs while we put in safeguards and precautions to make sure that if we are going to fund a lab in China, one that's known to potentially leak viruses, we need to make sure we hold them accountable for that we put in the proper safeguards and precautions so that we can continue collaborating with them again in the future and something like this doesn't happen again. And then in general, we should always be doing a reassessment of gain of function type research where we're taking viruses and we're making them more dangerous in a lab. Naturally, people, and I understand people say we shouldn't be doing that at all. Well, in a perfect world, we wouldn't need to have any nuclear weapons, but we don't live in a utopia. As long as there are other countries that are pointing their nuclear weapons at us, we have to protect ourselves. There's no way around that. I'd love to say that we could just stop doing this dangerous kind of research or not have to have any weapons to protect ourselves, but it's just human nature. We're imperfect beings, and we're always in conflict with one another. We're in conflict with ourselves, and that manifests in a larger context. So I don't know if that answered your question, but right. Yes, I do think it's possible. I do think there's a lot of circumstantial evidence pointing to a lab leak. I do think we should investigate it. I think we should remove the politics. That's not going to happen, but I think in a perfect world, we should remove politics from that. That's something that transcends politics. We're talking about a deadly virus that potentially escaped out of a lab, which is preventable. And regardless of you're a Democrat or Republican, you're equally exposed to that kind of a danger. And we should all have a shared interest in coming together and making sure it doesn't happen again.
Right. Well, I agree with you, Josh, in that NIH, was a short for the National Institute of Health, headed by Dr. Fauci, probably had good intentions. I mean, I'm sure they fund research all over the world, but why I think this has gotten politicized is a lot of people in the media, have idolized Dr. Fauci and NIH, and so a lot of journalists have actually, restrained themselves from questioning some of the science that's happened. I mean, not questioning the science of it, but the objectives, the public policy impacts of some of the scientific decisions. And so I agree with you that this probably, obviously you said there's not enough circumstantial evidence, but maybe this was something that the Wuhan Institute of Virology was just studying, and like you said, viruses are so small, it's so easy for them to spread, and it doesn't need to start with a lot of people. You can start with one person and literally replicate. And again, I don't know the demographic concentration of how the Wuhan, the Wuhan province. I don't know if it's a province or not. It's probably a city. I don't know, how condensed people live together. So that's probably something that I guess impacted this. But in general, how do we prevent this from happening? I mean, you've worked in a lab before, right? I mean, this is
I
scary
haven't.
stuff.
I haven't worked in a lab before.
You have not worked in the lab, but if how do we prevent this from happening? I mean, viruses are small, like, so scientifically, this is going to happen in the future. I mean, I feel like this is, as the world becomes more globalized, this is just the beginning of pandemics. It's only going to get worse from here. I mean, COVID-19, we're lucky it wasn't like Ebola or some of the other things that were worse. So how do we prevent this from happening again? That's like my last question to you.
Well, man, that's a tough question. And there's intuitively, I agree with you. I think that we're at the beginning stages of a new phase in technology, and we're probably going to see more biological mishaps occur. It's sort of like, going back to using nuclear weapons, when nukes became available, there's this amazing map. It's an animated map of the globe that it takes you through. It shows you all the nuclear bombs that have been exploded over like a course of 50 years. I don't know if you guys have seen that before, but it's pretty cool.
I have seen that, and it's actually insane. My professor showed it in one of my classes, and it's so I don't know, it's kind of mesmerizing to look at, to be honest with you.
Yeah. And you see in the beginning, it was like one bleep in the United States, one bleep in Russia, then a little bit more, then a little bit more. And then in the 40s and 50s, it was bombs going off all over. And then you start to see Pakistan and Iran, they got nukes. And then you start to see the UK, they got nukes. And you start seeing flashes there. And then Africa, and then Australia, and then the Pacific Islands. And then it started to calm down. It started to go back and recede. And now we haven't had a nuclear bomb explosion for quite a while. So I think, how did we get to that point? That's a tough thing to answer. We had to use them in war. A lot of people died from nuclear weapons. We had to go through the Cold War. We had to go through a lot of experiences to finally agree as a humanity to put some restrictions and limitations on the amount of nuclear research that we do and the amount of nuclear weapons that we stockpile. And so I think that we're likely headed in a sort of a similar fashion when it comes to biotechnological research, is that we are going to have to, as an international community, come together and agree on holding each other accountable, agree on oversight, and agree on harsh penalties for breaking those international agreed upon laws that we set for the kind of research we do and for mistakes, intentional or not, that happen. Because I would argue the stakes for biological mishaps, the stakes are much higher than with nuclear weapons. They're not localized like a nuclear weapon is. It can easily traverse the globe and infect everybody in a short amount of time. I will also just say that in the United States, if we do not find a way to come together as a country and overcome our political differences, that will be the downfall of our great country, because we need to be cooperating with one another in order to unify on this particular topic and to work together to prevent this. This is not something that should be politicized. This is talking about human health and welfare. That transcends your political differences. Unfortunately, we're going through a lot of different things at the same time. Journalism is changing. Mainstream journalism is, no one trusts it anymore. And so you have independent journalists out there. Everybody has competing interests. As you mentioned before, you have journalists that don't want to report certain things because they don't align with a political narrative that they're trying to push. And that happens all over the place. And also the access to information is so overwhelming. We're not evolved to consume as much information. And so we have so much information, it is impossible for the typical person to differentiate what's fact from fiction anymore. Even scientific, I would say even the politicization has infiltrated the academic research institutions as well, because you see papers that have a political lean to them to push a particular certain ideology in their publications, and that sours all of scientific research because you're no longer being objective. That's not what science is about. Science is saying we've observed this thing, we hypothesize it might be because of X, but we welcome you to challenge it with your own evidence and your own objections. Let's work together, putting our observations together to try to figure out what reality is and define it. It's not a game of I'm right and you're wrong. You have to prove it one way or the other. It's very much a collaboration, and our understanding of science and nature is constantly unfolding because we are advancing our technology constantly, and that opens new pathways for us to observe and discover new things.
I think that's a really important point that we've covered political polarization on this podcast a lot of times before, and again with this specific topic, I feel like you said, we shouldn't let our politics get into the objectivity of science because there's actually lives on the line. I mean, I feel like that was, that's why the pandemic got exacerbated because, certain politicians let their ego get in the way and politicize the actual science of it. And unfortunately, the whole world is kind of bearing the consequences, and we probably will bear the economic consequences for, the foreseeable future. But it was a really interesting conversation, Josh. We really value your perspective on this. And I think genomics is such a disruptive innovation force that it's only going to get better. There's going to be so many more people like you, who are going to examine these things in the spaces of their own homes. And that's a positive, in the short term. And as a society, we need to constantly, evaluate the ethical boundaries for this new technology.
Absolutely, man. It's a pleasure. Thanks for having me.
Appreciate it. And
Thank you, Josh.
Where can people find you? Make sure to plug your link.
The main place is Instagram. Check out Everyman Bio on Instagram. But also you can connect with me on LinkedIn. I'll connect with anybody. Josh McGinnis. Just look for me on LinkedIn, and then everymanbio.com on YouTube, Instagram, all the platforms. So
sounds good. Hey man, best of luck to you, and we'll make sure to plug your information in our description as well. Thank you for your so much.
Sounds good. Bye guys.
Josh.
All right.
See ya.